Okay. First of all, thank you very much, Professor Zenkova, to organize such a wonderful opportunity to get together. And this is the third Aquaphotomics international meeting. I'm very glad to participate in this opportunity. And probably some of you already noticed this Nobel Week dialogue. They pick up the topic. Water matters. This next week, December 10th, that's the Nobel ceremony is held. But one day before, they have the this kind of discussion about the water matters. I don't know who's going to participate in. Maybe some of you. Next week, going to Stockholm? Oh. Okay. But anyway, I'm one of the person who believes water molecule is most important for our body and for our health. And let me start. And I've been working on the aquaporin water channels for many years, more than 20 years already. And if we think about the role of waters in our body, I think two issues are quite important. First of all, every day we exchange our water. We drink water 1.5 liter or 2 liter, and also we take the water as a meal, and also we produce the water as a metabolite. And also we get rid of water by breathing or getting sweat, and also as a urine. And usually if we are healthy, water in out pass is very precisely regulated. But I'd like to point out to exchange the water is very important. Every day we exchanging the water. If you look at the cell level, even cells are always exchanging the water inside and outside of cells. I think this dynamic exchanging of water is very important. And second thing, we have the water flow in our body. If we think about water flow, probably you immediately imagine the circulation system. And also we have the lymphatic system. These two systems are quite important for our homeostasis. And if you look at cell membrane, there are a couple of ways for water to be permeated through the cell membrane. And the left side indicating the simple diffusion, water can pass through the cell membrane, of course. But the other way is the channel-mediated water movement through the membrane. And actually, aquaporin play a role in this case. And the important thing to have such kind of channel, of course, we can expect massive movement of water through the cell membrane, and more importantly, I believe, that's the regulation. We can regulate the water membrane water permeability. For example, when you get thirst, you have to concentrate on your urine. Or in other opportunity, if you drink lots of beer, maybe you will have right after, during the reception, then you have to get rid of lots of urine. So you have to dilute the urine. So the regulation of the water permeability is very important. You are not always crying. Tear formation is also regulated, and saliva formation as well. So having this kind of mechanism, we can regulate our body water dynamics. And many researchers try to identify what is the channel for the water. And eventually in 1992, Peter Agre's group at Johns Hopkins University in the United States, they eventually discovered the water channel, the protein, membrane protein, which functions as a water channel. And this is the egg, frog egg. They injected the cRNA coding this protein and transferred the oocyte from iso-osmotic media into hypo-osmotic media. The oocyte expressing this protein swelling very fast, and within a couple of minutes exploded. By this way, they proved this protein function as water channel. And because of this discovery, he won the Nobel Prize in Chemistry in 2003. And I will show you the molecular dynamics simulation movie based on the atomic structure of this aquaporin. And you can image how water passing through this channel. Here is the narrowest part of the pore. Only one water molecule is colored with yellow. So if you just follow this water molecule, it's easier for you to understand. And anyway, water can pass through this channel single file manner. I'm not going in detail, but this is, outside of the channel, water is moving around quite rapidly and always rotating, but inside the pore, I can show you once again, you will see the difference. And this is only, this video is constructed from the result of the calculation for just 10 nanoseconds. So actual movement of water is quite fast. 3 times 10 to 9 water molecules can pass through this channel per second. So you can imagine how fast it is. To establish the new field, water biology, we are now concentrated on the 3 major topics. One, regulation of body water dynamics by aquaporins, and second, we try to visualize a water molecule by live imaging using nonlinear multipolar microscopy. And third, we try to understand the behavior of the water molecules by aquaphotomics, established by Professor Zenkova. And I just came here a couple of hours ago, but I think the Dishan Koichi and Saya Tanaka from our group, they already presented our work regarding the aquaphotomics yesterday or this morning, so I don't talk about this third one. And first, I just to briefly introduce you our live imaging method of water molecules. And next, I just focus on one of the aquaporins, which is the brain aquaporin, aquaporin 4. And we introduced the Raman scattering, coherent anti-Stokes Raman scattering method, so we can visualize water molecule based on the water molecule vibration. And using the features the Raman, the difference of the feature of the Raman between heavy water and water, we can distinguish the water molecule from the heavy water. Water molecule, coherent Raman peaks here, but heavy water is here. So we can clearly distinguish water from the heavy water. So what we did, we just culture cells with the water and rapidly exchange the extracellular solution to heavy water. Then monitor by gas microscopy. And this is the time, 0 milliseconds. So almost every 30 milliseconds, we can monitor. So here, you will see the heavy water. Now we are visualizing the water. So if there is a heavy water, get dark. First, of course, the extracellular solution replaced by the heavy water, and already started the exchange between water, inside the water, and the extracellular heavy water through the cell membrane. And by within 1 second, almost they reach to the plateau. And if we perform the line scan, we can even increase the time resolution. So this is the cast intensity. The outside, of course, immediately change, getting dark. Cast signal is decreased. But if we don't have the aquaporin on the cell membrane, it's much slower. And if we have the aquaporin line with red, much faster, of course slower than the extracellular solution. But by this method, we can clearly distinguish the simple diffusion from the aquaporin-mediated water movement. And we also apply this method to the cyst. And we succeeded to measure the water permeability, diffusion of water permeability differently between apical membrane and basolateral membrane. And also, for example, this is also the cyst, and if we, from the collecting duct of the kidney, if we, here is the aquaporin 2 distribution, if we treat the cyst with antidiuretic hormone vasopressin, aquaporin 2 move to the apical side, you see. Then we also measure the water permeability, diffusion of water permeability, apical and basolateral separately, and we can reproduce the increase of water permeability only in the apical luminal side, but not basolateral side. And we now apply this to the tissue. I mean the isolated, in this case, the pancreatic duct. We microdissected the pancreatic duct from the mouse and performed the double perfusion. So inside the tube and outside the duct, we can manipulate differently. So we can exchange the water and heavy water either inside or outside. Then monitor this curve. I don't present the data, but it's now possible. Next stage, we're going to try to apply this CAS method to the brain tissue. This is just started, but so far we think it's feasible. And then I'm going to talk about the aquaporin water channel in the mammalian. Including human, we have 13 different aquaporins. Aquaporin, for example, aquaporin 0 is only expressed in your eye. So if this aquaporin 0 doesn't function, you will develop the cataract. And aquaporin 1 mainly expressed in the endothelial cells of your capillary. In many tissues, and of course expressed in the kidney as well. And aquaporin 2, I just introduced to you. This is very important aquaporin, kidney aquaporin, to concentrate of your urine. And aquaporin 3 in the skin and so on. Aquaporin 5 play a role in secreting tear or saliva. And I'm going to just focus on this aquaporin 4, which is abundantly expressed in the brain. And here, this cartoon indicating the distribution of aquaporin 4. Some of you are not so familiar with the brain anatomy. We have mainly two types of cells. One is neuron, and the other one, glial cells. Among glial cells, astrocytes play quite important role. Almost every synapse, I mean connecting the neurons, surrounded by the astrocyte process, and also all the brain capillary or a small vein or artery are surrounded by astrocyte process, making the blood-brain barrier. And also, here is the ventricles. We have the ependymal cells, and astrocyte one arm reach to these ependymal cells. And also, this is the surface of brain, pia mater. Pia mater also astrocyte process reach to this pia mater, and aquaporin distributed here as well. So by just seeing this pattern of the distribution of aquaporin 4, you may imagine aquaporin 4 play an important role to regulate the brain water dynamics, especially the interface of the brain parenchyma and cerebral spinal fluid or systemic circulation. But honestly, we still don't know the physiological relevance of this aquaporin. But in case of brain edema, this aquaporin plays a role. For example, in this case, we introduced the water intoxication and comparing the wild-type mouse and the aquaporin for knockout mouse. Aquaporin for knockout mouse has a much better survival rate, indicating if we have the aquaporin, in case of developing the brain edema, that is not good. So many groups all over the world, including ourselves, try to develop the inhibitor of this aquaporin for the treatment of brain edema. But we still don't know the physiological relevance of this aquaporin. But very interesting concept, new idea came out 5 years ago. And back to the lymphatic system, you may notice we have the lymphatic capillary whole body, any kind of peripheral tissues. But if you look at the brain, there is no lymphatic capillary. So brain should have very different lymphatic system. And 5 years ago, Professor Nedergaard group, she's now in Denmark, proposed very interesting concept. What she said, published in the Science, what she said, there is the convective flow in the brain parenchyma, especially from artery side of vein side. And aquaporin 4 play a role in creating this flow and maintaining this flow. But, and also 2 years later, the other group found there is some lymphatic vessel in the meningeal membrane, I mean the outside of the brain parenchyma. But still, lots of things are missing, and this concept is also very controversial. We still don't know if this is true or not. But concept itself is very attractive. And they also showed, for example, if they inject the radioactive amyloid beta accumulation is always seen in the Alzheimer disease patient. So clearance of amyloid beta is very important to keep our brain function. So if they inject the amyloid beta, then they monitor the clearance of the amyloid beta by comparison between wild-type mouse and aquaporin 4 knockout mouse. And as you see here, aquaporin 4 knockout mouse has much delayed amyloid beta clearance. And ourselves, we also performed injecting the, some dye into the CSF and compared between wild type and knockout. And knockout, the dye distribution is quite limited compared to the wild type. This is just some of the indication, but we still don't know what's the role of aquaporin in this brain lymphatic system. Sorry, some of the characters in Japanese, but our working hypothesis, if aquaporin 4 play a role to get rid of amyloid beta, maybe that play a role to developing the pathophysiology of Alzheimer's diseases. There is the familiar Alzheimer's disease patients, then we already know which genes are mutated in these familiar Alzheimer's disease patients. We introduced that mutation into mice, so making the transgenic mice expressing the mutated genes. But that transgenic Alzheimer model itself, unfortunately, doesn't reveal any severe phenotype as Alzheimer's diseases. And I didn't mention, but aquaporins for knockout mouse, if we don't give them any challenge, there is no obvious phenotype. There is some hearing loss, but that's it. So our working hypothesis, if we have create Alzheimer transgenic mice based on the aquaporin for knockout by mating, we may see the more severe phenotype. And it was the case. Here we monitor the mouse for 24 hours by video camera. As you may know, mice they sleep during the daytime. So this is the daytime and with light. And when it gets dark, they start to move around. And here is the three kinds of mice. One is the aquaporin for knockout with blue, and green, Alzheimer's disease transgenic mouse, and red, the double transgenic mice. So even at this stage, by our eye, we didn't see any difference. But by introducing this 24 hours monitoring, already this double knockout, double transgenic, has a little bit less movement. This is at age 20 weeks. And 1 week later, you will see more distinct difference in the behavior among these mice, and 28 weeks even more, and 32 weeks. By this age, we may feel something different, but not like this kind of significant difference. And further, after 36 weeks, at this age, everybody can distinguish. Here there are 4 mice. One is the wild type, one aquaporin for knockout, and one Alzheimer's disease model mouse, and last one is the double transgenic. I will show the movie. You see. You know, I'm pretty sure everybody agrees. One mouse is very different from the other 3. This is the actually double transgenic mouse. And they also reveals the seizure. More than 40% of Alzheimer's patients, they have the epilepsy. So we still don't know the role of aquaporin 4 in the brain lymphatic system, but definitely plays some role to development of the Alzheimer's disease pathophysiology. And another attractive thing of this Nedergaard hypothesis, they showed this brain lymphatic flow is regulated by sleep. During sleep, this flow is activated compared to the daytime. I mean, in this case of mice, so daytime they are sleeping. So anyway, by sleeping or anesthesia, this flow is activated. We still don't know why we have to sleep in detail. But one of the answers, if this is correct, one of the answers is we need sleep for clearance of the brain. So during the sleep, we get rid of the waste of the metabolites. We still don't know this is the true or not. But by 5 years ago, I thought quality of sleep is very important anyway for our health, for our healthy aging. Usually, when we get old, the sleep levels get more superficial compared to the child. So 5 years ago, I started collaborating with the Professor Mitsukura at the Science and Technology University. I believe she gave a talk on Sunday or Monday, Sunday, yeah. And we are succeeded the monitoring the accurately the sleep stage without using EEG or PSD polysomnography. Usually you can get many watch or any kind of monitors. They say they can assess your sleep level. But as far as I know, most of them, the accuracy is only 50% or 60%. Our method is reached to the 80 to 90%. So we recently submitted the patent and just established the startup company with her. The name of the company is eLife. So this is a group, and some of the data I presented today in collaboration with the Santry. We have the Seimei Miguru project, joint research project. Thank you for your attention.
Thank you very much, Yasui Sensei, for this very interesting talk about aquaporins. I would just like to point out one thing that I missed to say about Professor Yasui. He is now Vice President of our newly established Aquaphotomics International Society. So, congratulations. So, anyone has any questions? Okay, well, I can break the ice, or you think about it. I was interested in, there are different types of aquaporins in human body.
Yes.
But what is the difference in the structure and how?
Actually, I skipped it. First of all, we can categorize the aquaporins family into two main groups. One group we call classical aquaporin. They are only permeated by water molecules. But the other group we call aquaglyceroporins. We call they are permeated by glycerol or rare or some other small non-charged molecules. And here is based on the atomic structure. If you compare the pure aquaporin, classical aquaporin, and aquaglyceroporin, actually this group has a little bit wider, narrowest part of the pore, constricted region. Actually, 1 angstrom larger than this. So this kind of difference may create different permeability characters. But I don't have the slide. But anyway, these groups are quite similar to each other. The only difference is the regulatory region. Usually the end of the N-terminal or C-terminal, the proteins, they interact with other proteins and so on. So in terms of regulation, for example, aquaporin 2 regulatory mechanism has been well established. For example, phosphorylation of the C-terminal region of this aquaporin 2 is very important for the regulation. So why we have to have this kind of these numbers of aquaporins? One of the answers is first of all the little difference in selectivities, and the other one is in terms of regulation.
Thank you. And one more question from me, please. So I saw that you tried to establish the link between sort of like aquaporins and amyloid beta as sort of like a main, let's say, biomolecule, which is considered to be important for development of this disease. But there are other contributors like tau protein, or for example, there is a decrease in
actually, we compare the accumulation of amyloid beta between the Alzheimer mouse model and double transgenic. As I showed you, that there is a significant difference in the behavior, but in terms of amyloid beta accumulation or tau phosphorylation, there is no significant difference. That was big surprise for us. But probably unknown molecules, other unknown mechanisms are working on this. So we are now trying to identify such a new molecule or new phenomenon to get these mouse worse. In addition to the amyloid beta accumulation or tau phosphorylation.
Thank you. Very interesting. Thank you. Okay, Professor Tsenko.
I was listening to the talk and this development on Alzheimer, and very interesting, and it just appeared to me. When we worked with prion protein, and we monitored 2 years ago, already many years ago, through the brain and through the gut, we were able to diagnose prion disease earlier from the gut, not the brain. So do you think that probably that there is something related to Alzheimer coming from the gut?
Yeah, of course. For example, the Nobel laureate Tanaka sensei and Shimazu, they recently they established the method to measure the amyloid beta in the blood, only one drop of blood. So now they try to introduce this method to monitoring the very early stage of the Alzheimer's diseases. But we don't have to measure the beta amyloid, but I agree with you. We may detect something different even in the body or systemic saturation as well.
Yeah, thank you.
More questions? Okay. Thank you very much, Professor Yasui.
Thank you.